Hello, my name is Mandisa Holmes, and I'll be talking to you all about 3D cell culture. Let's get into what 3D cell culture is. 3D cell culture is an environment that allows for cells to grow and interact with the surroundings in all three dimensions. The main reason for using 3D cell culture for growth is simply because it's a way better way to represent human tissue outside the body. 3D cell culturing became of severe importance in the 1980s when Mia Bassel, a researcher, proposed the importance of tissue microenvironment and 3D culturing technique for cancer research. All 3D cell cultures can be grown with either a supporting scaffold or no supporting scaffold. And we will discuss the two as well as others. When 3D cell cultures are cultured within a supporting scaffold and allowed to grow in all directions, it is considered to be a scaffold 3D cell culture. The most common of the scaffold 3D cell cultures are hydrogels and inert matrices. Hydrogels can derive from animals or plants or even synthesized from chemicals, whereas the inert matrices are made up of polystyrene. Now, 3D cultures that can be grown without the presence of support of scaffolds are considered a scaffold-free 3D cell culture. Scaffold-free 3D cell cultures are low adhesion plates, micro-pattern surfaces, and hanging drop. The next is organoid. They are self-organizing 3D tissue cultures that have been derived from stem cells. And the last being microfluidic organ on a chip model, which is a cell culture chip that is engineered to imitate the physiology of an organ. Some of this may sound a little familiar to you, as we did discuss cell culture in Module 5. So after that explanation of what 3D cell culture is, you might be asking yourself, so what exactly is it used for? Well, let me tell you. 3D cell culturing can be used for drug discovery and drug therapeutics. 3D cell culture can be used as a cost-efficient screening platform. 3D cell culture is also used for screening for small molecule drugs, understanding disease pathways, predicting the efficiency or toxicity of drugs, cancer research, due to the ability to replicate the body's natural environment, stem cell research, and disease mechanisms, just to name a few. Just like any Apple product, just when you think you have the best version, a newer model comes out. That seems to be the case with 3D cell culture. Before 3D cell culture, there was 2D cell culture. So oftentimes 3D cell culture is compared to such. So I'll join in and do the same. Some of the limitations include not being able to get oxygen and other essential nutrients to the right place, which essentially happens when larger cultures have the challenge of distributing the nutrients to the right place, which only essentially happens in larger cultures. Other limitations include having unwanted elements within the culture, throughput, and difficulties measuring and viewing the culture. Some of the advantages include the fact that 3D cell cultures mimic the tissue and organ structures. There's good reproducibility. There's an incorporation of fluid flow, and it ultimately reduces the use of animal models. Let's get into some controls. Of course, the obvious control here would be in vivo, basically having an animal cell that would support the growth of the cell. The only thing with that is it takes from why we use 3D cell culture, which is being able to grow a cell outside of an animal or a human host. So the next control we would get into would obviously be a negative or a positive control, which would mean having cells that are not being treated and comparing those to cells that are being treated, making sure the environment for both are the same. This first figure is from a paper entitled 3D Cell Culture Systems, Tumor Application Advantages and Disadvantages. Now this figure represents different synthetic strategies of 3D matrix bases. We have collagen, lyophilization, electrospiding, stereolithography, and microfluid. Porous material can be seen in A, as it shows the discrimination against the 3D models of the ECM of nanoporous scaffolding. In 3B, you can see that the lipolization is a dehydration technique, and that results in the formation of the interconnected circular oval porous microstructure. 3C is electrospinning hydrogel. Because the electrospinning technique uses electrical forces to form a network of fiber, it forms polymer solutions or melt. 3D is 3D printing scaffolding for 3D cell cultures via stereolithography. This has different layers that harden to visible light or infrared. 3E is seen as the microfluid. This next figure is taken from a manuscript entitled Mucus-Producing 3D Cell Culture Models. This manuscript basically talks about 3D models that contain mucus-producing cells, simply because they play a vital role in drug absorption. This figure represents the upper respiratory tract mucosa in different 3D cell-based models. A is the representation of the human nasal mucosa, which contains mucosa-producing goblet cells, ciliated cells, basal cells, cilia, mucus, and submucosa glands. Both B and C are 3D non-immortalized mucociliary cell-based models. And D is the illustration of the EMC incorporating microfluidic device, which is composed of human nasal epithelial cells and human microvascular endothelial cells, which in turn create a microenvironment similar to having the native nasal mucosa. This is a figure that I got from the manuscript 3D Cell Culture Models, Drug Pharmacokinetics, Safety Assessments, and Regulatory Considerations. This figure shows the numerous developments of 3D cell culture systems. In this figure, the 3D cell culture systems are being developed to mimic the physiological organs, just as we discussed earlier, which in turn helps the body evaluate the drug absorption, how it's distributed in the body, how it's excreted, the metabolism of the drug, the function of the heart and how it reacts on the heart, as well as the immune response to the lymphatic system. The manuscript states that the goal is to integrate these organ-specific 3D models into a multiple interconnected microphysiological system. And with the integration of such, it would allow for the evaluation of the administration of drugs on the pharmacokinetics and ultimately improve drug safety altogether. Thank you for listening to my presentation. Thank you for your time, and hopefully you're walking away with a little piece of information.